genomics. There's lots of other kinds of computational science that happens in biology, and I'll mostly talk about one application problem so you can get some idea of what the algorithms look like, but hopefully you'll get a flavor of the general field. So just a little bit about the lab. I don't know how much you've said about Lawrence Berkeley Lab, but as Phil said, both of us, our day jobs are up there, and the lab does a lot of science in a lot of different areas. It also, one of the things the national labs do in general is they run big user facilities. So these are some of the user facilities at the lab. And I'll say a little bit more about NERSC, which is one of the facilities that is in the Computing Sciences Organization. It's a supercomputing center. If you take CS267 with me in the spring, you'll be using the NERSC supercomputers. And I'll also say a little bit about the Joint Genome Institute, where they do a lot of research in genomics. And the Lawrence Berkeley Lab is a Department of Energy lab. So just a little bit more about the computing area so you know a little bit about who I am. So I run the Computing Sciences Organization, but I also do research in computer science. So in some sense, I work for myself in that org chart, which is very convenient. But I run, as we said, the supercomputing facility. We also run the wide area network, and then there's a research division which does applied math, or the kind of things that Phil has talked about, computer science, data science, and we have a lot of partnerships with other science disciplines, including biology. So if you were following what I said about this being a Department of Energy lab, you may wonder, why does the Department of Energy care about biology? And so I'll just give you a little idea of what things happen at the Joint Genome Institute. This was a slide put together by somebody from JGI about the kind of things that they do. So JGI was actually established because they were involved in the original Human Genome Project. So they're involved in the sequencing of the human genome, one of the two main organizations that did that. And that was partly because it was such a big project that people recognized that the labs are one of the kinds of places where you can attack really big problems like this. And so that was why JGI was established as this big center to do the Human Genome Project. But DOE doesn't really work on humans. That's NIH's job for health applications, a little bit of NSF's job. And so, but it turns out that there's still a lot of biology that's related to energy. So what they do today is they worry about things like bioenergy. Can we grow, say, certain kinds of switchgrass that we can use for biofuels? Carbon cycling, so trying to figure out whether, for example, the microbes, which is what we'll be talking about later, are going to absorb more carbon. So are there these little species that are going to either release or absorb carbon? Biogeochemistry then is looking at sort of the chemistry of how all the biology that's out there in the environment works together with and produces chemistry and is influenced by it. So what they tend to do is the biology of plants, of fungi, of metagenomes, which are communities of microbes, individual microbes, and then engineering biology, which is, for example, synthetic biology. So using biology to produce some kind of a chemical that you're interested in, maybe, although not the application domain here, one of which you're maybe familiar with would be antibiotics. So using some biology to produce antibiotics or some other kind of chemical. But engineering the biology to produce exactly what you want. So they have a lot of sequencers there, and they do a lot of computational analysis and things like that. And just to give you an idea of what they're computing, okay. So microbial analysis is the main application. It is of interest to the Department of Energy. It's also of interest to the NIH. If you've read any of the articles about your human microbiome, you know that your gut microbiome is really important, and your skin microbiome and everything else are really important in human health. And they affect all kinds of things in your life that we haven't really understood until recently. The same thing is true in the environment. So if you're a farmer and you're trying to figure out how to make your soil really rich for growing a certain crop, the microbial species, the bacteria and so on that live in the soil are really important to understanding that. And for example, understanding how drought tolerant your field, your region might be. So these are some of the applications that I mentioned before, but specifically this is about understanding microbes, which are single cell organisms such as bacteria and viruses. And these microbes tend to not live in isolation, they live in communities. And so one of the problems is, and the comparison to say the human genome is, although that was a big problem, the original sequencing and analysis of the human genome was a big problem. It was somewhat simpler because you take, say, a blood sample, and that has, that should just have your DNA in it. So it has multiple copies of that DNA in each one of the cells, but it just has your DNA in it. When you pick up a sample of soil, it might have 1,000 species in it all mixed together. And so you send that into the sequencer, and what you get, by the way, the sequencers, what they produce are little fragments of DNA. They don't produce your whole chromosomes. They don't just sort of read the chromosome out. They produce little fragments. So now the problem is that when we're studying these microbes, we're studying actually communities of microbes altogether, and we're going to have to figure out how to analyze that data. Some of these have never, they can't be cultured in the lab, so you can't really grow them in isolation. Some of them, one of my colleagues likes to say, would you like to come and discover new forms of life? Because you're actually discovering new forms of life that we haven't ever identified before, because there's these little microbial species, and when you sequence them, you can say, oh, there's a new form of life that we haven't seen before. So, and metagenomics is the problem of analyzing these data sets that come from these microbial genomes, these microbial communities. So you'll have them all mixed together. Okay, it's a big data problem. This is the obligatory graph about the growth of these data rates. This is specifically metagenomes. So there's similar graphs that you would see on the cost of sequencing the human genome has come way down to the fact that you can get your genome sequenced for about $1,000. Now this is looking though at these kind of metagenome samples, which you might think are not of so much interest, but there's actually a lot of interest in it. This is the number of terabases. So we're going to count things a lot in a base, which would be a character, ACTG. They tend to come in pairs because of the way biology works, but anyway, we'll look at those. And so this is an open database called SRA, and the size of that database grows roughly every 11 months, and it has both some internal stuff and the part that's open to the public. Okay, so how hard is this problem of metagenome assembly? The problem, as I'll talk about in more detail, is taking these little fragments of DNA and trying to reconstruct them into a complete genome, or multiple genomes in the case of the metagenome. So this graph shows, and this is a little bit old, but I think it still roughly tells the right picture, is if we were trying to sequence one of these metagenome data sets, and there's different groups of them, the hardest ones being soil. So so the x-axis is the total amount of data that you sequenced for this particular problem, and the y-axis is the percentage of those reads. They're called, the things that come out of the sequencer are called reads. The percent of those reads that actually could be mapped back to the solution, to your assembled solution. So your computer program is going to take the reads and try to produce something that's approximating the genome for each species, and this says once you're done with that, you go back and try to figure out which of the reads actually are represented in that data set, and it's under 10% of them for the most part. And as you can see, that even though if you sequence more and more data, we're improving it a little bit, but we're not really getting very high percentages. So one of our goals is to get much larger percentages of the genes assembled, and of the reads assembled into the sequences, and to work on these really hard data sets such as soil. We actually did, and it's not in my slides, but we did a run last week on a big environmental sample, and got 34% of the reads were able to be mapped back into the solution, and so we're very encouraged by that. It's sort of a preliminary result, so we need to analyze it to make sure it's kind of legit in various ways. Okay, so I'm going to talk about the short read de novo genome assembly problem in more detail to give you an idea of what the computing aspects and the algorithms look like. So to take that apart, there's a bunch of different kinds of sequencing technology. The sequencers that are the cheapest and the most popular kind of in production are called the Illumina sequencers. They produce fairly short fragments, about 100 base pairs, so about 100 characters long, maybe 100 to 200 characters long. And so those are the things that we're going to work on for assembly. There are other kinds of sequencers, such as PacBio and the Oxford Nanopore, which is a little assembler. They're about the size of a memory stick, and you can kind of plug it into your computer and put something on it. Anyway, some of these produce much longer strands of DNA, which is a good thing for assembly. The problem is that right now, at least, those longer assemblers, first of all, they're more expensive. Secondly, they tend to be slower. And thirdly, they produce more errors. So all of these things produce errors, which is one of the things that's going to cause problems with our analysis. And the rate at which we will have a percent error, maybe 1% error or so in the Illumina data, the short reads that I'm going to talk about. The long reads, and I have there's a couple students working on that problem as well, along with Aiden Bulich. But the long read problem is they produce about 15% of those of the individual characters are errors in the PacBio data. And so the question of how you put that back in together into a correct assembly is much harder. It's much easier because they're longer, but much harder because of the errors. Some other things, and I may talk about this, protein clustering. We run big, huge sparse matrix problems in order to figure out what proteins are related to what other proteins, and the proteins really correspond to genes, so you can think of that as gene clustering. And sometimes we're looking at the relationship between things, other big sparse matrix problems, which I probably won't have a chance to talk about today. But so I'm just going to give you a flavor of really the genomic analysis problem itself, but just to point out there are these other kinds of sort of machine learning type of applications that come up in analyzing biological data. Okay, so what's this metagenome assembly problem for short reads? Okay, so it's like this puzzle problem. You've got data that has, imagine that you've been given 1,000 puzzles, and fortunately you're going to have multiple copies of the puzzles because each one of them has some of the pieces missing, and each one of them, by the way, has some erroneous pieces that aren't going to appear in the final puzzle thrown into the mix. But you take those 1,000 puzzles and maybe about 20 copies of each one, and you throw all the pieces together, and you don't get to look at the cover of the puzzle, right, that shows you what the 1,000 images are that you're trying to build, because it's de novo assembly. So that's what de novo means, that we're building it from scratch. That was what the Human Genome Project did the first time for the human genome, which is they said we're from scratch trying to figure out how do all of the pieces, all the genes, the DNA sequences fit together in the human genome. Once they have that, and the way most medical applications are run when you're analyzing, if you go and get your DNA sampled, is they take all of your little puzzle pieces and they say, oh, this one looks like it fits right here in the reference genome, because even though your DNA is a little bit different than everybody else's DNA, there's enough similarity that you can kind of match up the pieces. So it's kind of like, for the human case, we have the cover of the puzzle that we can use as a reference for putting it together. And but for these metagenomes, we don't have any pictures. We don't know what we're looking for. Okay, so what does this look like? So as I said, they produce errors in this. So here's an example to make it a little bit more concrete. This is a very small problem, but I've got 4 reads that are coming in. So these are my strings of DNA data. And they're, the red characters that you can see there are supposed to indicate that there are errors. So there are characters that can be, that are flipped. So that G up there should be a C, because all the other copies of this. So we're going to read the genome multiple times in order to get rid of the errors. So that's going to be the basic trick, is to read it, say, in the human case, we'd read it 20 times, and then we can have pretty high confidence we can get rid of most of the errors. So you can have things that are flipped, which we call substitution. You can have things that are inserts, so an extra character inserted into it, or you can have things that are dropped, so things that are missing. So deletions, insertions, and mismatches are the basic kinds of errors. But the idea is that because we have multiple copies, we're going to sort of figure out what the consensus is about what these strings actually look like and produce a longer string of DNA. You can't really tell it's longer from this picture, but that's also, it's supposed to be longer. Okay, and we're trying to get out things that are, say, tens of thousands of base pairs long in the output, so that the point is to be able to find genes. And genes are going to be longer than those input sequences, so we can't just look at the reads to find the genes. But once we get these longer fragments, even if we don't have complete genome, we don't have like a complete chromosome from beginning to end, they'll be long enough that we'll be able to find a lot of the genes in it. Okay, so this is just, I think this is kind of what I said before. Soil is a lot harder than some of the other kinds of metagenomes, and the JGI people are very interested in that. They're also interested, they don't really do mammals. Not only they do not do humans, they don't tend to do mammals, but they do cow rumen, they're basically their microbiome, because they're trying to figure out how cows break down grass, so they can figure out if they can use that to break down switchgrass for biofuels. Okay. So what does the computation look like? There's 4 phases. I'll probably talk about the first well, one or two of the middle ones in a little bit more detail, and I won't say so much about the others. But just to give you an idea, we've got the reads that are coming in. So these sequences of DNA, and they may have errors in them. What we're going to do first is we're going to chop them into what are called k-mers. And k-mers are going to be even smaller fixed length pieces, smaller pieces, but they're all going to have the same length. And that's a little bit counterintuitive to me, that the first thing you do is you're trying to make them longer, but you actually chop them into shorter pieces. And when we chop them into shorter k-mers, we're going to do it in a sliding window fashion. So if we pick a k-mer length, and I'll show this on the next slide, so maybe I'll talk about it there, we're going to do a k-mer, we're going to compute a k-mer at every position. We're then going to take those k-mers and we're going to analyze them in such a way that they look like a graph, and we'll build contigs out of them, what are called contigs, so longer fragments. These are at this point we will have fairly high confidence in what we've assembled. We'll then go back, because those aren't long enough, we'll go back and look at our reads and try to map those onto the contigs and see if we can glue together some of the contigs and say, well, there's a gap in here that we weren't able to get from this initial analysis, but we can tell that these 4 or 5 reads all overlap with it. Maybe we can just narrowly look at that region and figure out how to fill it in. So we do some gap closing there. Okay. So the k-mer analysis problem is basically a big histogramming problem. Have you talked about histograms in here? Probably not. Okay, so histogramming, you're just counting things. So simple histogram, given all the words in a book, put them into 26 buckets, each of which has, how many of those words start with the letter A, how many start with the letter B, how many start with the letter C, and so on. And we'll just count them. We'll say this book has, whatever, 953 that start with E, and only 2 that start with Z or whatever. So it's just counting the number. So why are we going to do that? So we're going to do that here. Let me first of all tell you what the k-mers are. So we start with these reads, and a k-mer is going to be a fixed length string, and you'll see later why we want it to be a fixed length string, but for right now, just take my word for it. A typical length in practice is going to be 51. There's reasons why you want it to be odd, having to do with the way that there's symmetry in the DNA, but anyway, 51 is typically used for something like the human genome, and even in a lot of the metagenomes that we're using. In fact, in metagenomes, we'll actually try a few different lengths, and so we'll iterate over different lengths of k-mers. Because they have different trade-offs. But we're going to look through these strings and chop it into these k-mers. Now it's a sliding window in the following sense. So if I've got this read, GCTA, et cetera, and then all the formers in it, so we wouldn't use such a small value, but here just to show graphically what we're doing is we're showing each 4-character string that starts at every single position in that original read. Okay. So what we're going to do is, now we've got these k-mers, these, in this case, formers, we're going to throw them all into a histogram, and we're going to count how many there are of each one of those. And that's across the entire genome, the entire data set that we've read. Remember, it's got genomes all mixed together from different species and so on, but we're going to throw it into a hash table and basically, into a histogram. I'll tell you about a hash table in a minute. We'll histogram them and count how many there are. If there's only one instance of a k-mer, we'll assume that that has an error in it, because we're going to try to read the genome multiple times in order to make it possible to reconstruct it. And if we say if it only appears once, it's probably an error. There was some bit that got flipped or whatever in the sequencer. You can set that threshold a little higher. You can say if I don't have at least 2, or at least 3 or 4 or whatever, but somewhere around a very low number. We're going to just immediately throw out k-mers that don't occur more than once. Yes?
In some typical sequence, chromosome, so there are 20 to the 4th, 4 to the 4th formers, right?
Right.
Okay.
Right.
So that doesn't seem like
it's
such a large number that we wouldn't.
Yeah, right. So that's why we won't use a k-mer value of 4. We'll use a k-mer value of 51, so that we actually have a large number of possible k-mers, right? And so then the chance that you're accidentally going to see a k-mer so the problem with formers is you're going to see the same former, like even whatever. You're going to see that multiple times throughout your genome, and everything is going to occur multiple times. You won't be able to throw anything out. But if you pick a big enough number like 51, then
I'm sorry. I thought I misinterpreted the 4
Right. The 4 is just like the example for the slide, and the 51 is what you'll actually use. So it's 51 to the, 4 to the 51, right? So now it's a much bigger number of possible values. And so we do have to be, by the way, a little careful about how we histogram those, because you also don't want to build a table of size 4 to the 51 in order to count these things. But there's a trick that we'll use called a Bloom filter in order to make the counting a little bit more tractable. That's a table that's somewhat smaller than the 4 to the 51 size table, and it's got a sort of statistical property that is related to hashing, which I'm going to talk about later. So let me not try to explain a Bloom filter right now. Remind me when we talk about hashes if you want me to tell about it. And I tried to take the parallel computing stuff out of here. You can see this lecture again next semester if you want. We'll talk more about the parallel computing. Yes?
Sorry, maybe I missed it, but why 51?
Why 51? So it can be an odd number, and that has to do with the fact that there's sort of you get some symmetry, and you'll see these over again. And but I would say 51. So it's sort of the Goldilocks. It's sort of big enough that you don't have this problem that you're going to see things over and over again. These repeated that you'll have too small a number, and everything will look like it occurs in the genome. And so that if it's too small, it's not good. If it's too large, what's the problem? Anybody have any sense of what's wrong with a K-mer value that's too large?
You get a lot of values that just have 1 or 0 counts.
You will, but you also will. Most of your sequences will have an error. With an error rate of 1%. Right? If you take 100 MERS, right, statistically you're going to have an error in each one, and even at 50 you're kind of getting up where you're going to have accidental errors in there. And so you're going to end up throwing those out, even though they have good data in them. There's some place there's an error, but there's some good data. So you want to make it short enough that you're going to find a complete K-mer that has no errors in it, but long enough that you're not sort of accidentally, that you're actually able to tell things apart. And the other big problem that you have when they get too short is what are called repeated regions of the genome. So in the actual genome, there are things like whatever, GCTA that appear at multiple places. And if you pick that, as we get to the later stages of the analysis, everything's going to get tangled up because we're not going to be able to tell, does that one appear here or here? We think it's the same. It'll make it look like the whole chromosome is kind of wrapped around itself. And so you want them long enough that you can disambiguate the repeated regions.
Is that just like sorceress, like trying it a bunch of times and determining 51 is right, or can you actually like derive this
So in practice, I would say I think that came from experience. There is statistical analysis on it to say, for a given read rate, for a given error rate. And by the way, the errors in the Illumina data do not appear randomly. The errors in the PacBio data do appear randomly. So you do need to have some model of what the error rate of the sequencers looks like and what its distribution is, and then you can do a statistical analysis. But and we are doing that in some of the other work to try to figure out what's the right K-mer length. But yeah.
And why do you think that some sequence that only has 1 count is going to be, or a low number of counts, is an error necessarily, not just a quirky thing?
Because we're going to read our genome multiple times. So we're going to take the data and we're going to send it through the sequencer essentially, 20 times. And so from that, we think with high probability you should at least have 10 copies or whatever of that. And so anything that, if you read it enough times, then you can throw out the low ones. Yeah, good question. All great questions. Any other? Okay. So so that's what we're doing. So what there's another thing I haven't really talked about, but will come up later, is what we're doing from this is we're finding high quality K-mers, right? So these fixed length things, 51 characters, all of the ones after we've put them into this histogram, we've counted them, we've thrown out the ones that don't occur frequently enough. What we've got are good K-mers left, and they may occur many times in the genome or whatever, or at least in our read set. But we've got a copy of each one. We are also at this point going to compute what's called the left and right extension of that K-mer, which is in the original read. Let's say this K-mer TACG. Well, to the left of the T was a C. And to the right of the G was another G. And so I'm going to remember, I've got a K-mer of length 51, but I'm also going to remember that when I read it originally, it had something to the left and something to the right. Okay? And what I'm also going to do is throw out any k-mers that do not have unique extensions. That is, if I've got 2 copies of this k-mer and one of them says, oh, the next character is a C, and the other one says the next character is a T, then you'll say, sorry, but I don't know what to do about that because I know chromosomes don't have branches in them, so I'm going to just throw them both out. And so that's going to cause us to not get very long sequences, and we'll come back later and try to fix that. But remember the left and right extensions because it'll come back up in the next step. Okay, so the next step is what's called contig generation. We're going to take those k-mers that we had computed. Imagine that we filtered out the errors through that process. And we are going to remember the left and right extensions. And we're going to use that set of k-mers to build these longer contigs. So what we do is we are going to take all of these and we're going to store them in a hash table. And I'm going to tell you on the next few slides how hash tables work, but they're a magical data structure that makes things run really fast if you've got the right kind of data for putting them in. And we're going to store in them a k-mer like this AGTC, and I'm going to store the left and the right extension as values of that k-mer. So what the hash table is going to let me do is, given a k-mer, look it up and tell me what was the left extension and what was the right extension. Okay, so that's going to be the idea of this data structure. And then we may have to come back to this slide because we're going to think of this hash table as a graph. But let's do that after we talk about what a hash table is. Okay, so hash tables come up all over in a lot of applications you use every day, Google search and things like this. And so the easiest way to think about it is I'm trying to build a dictionary. So in this case, the words are going to be k-mers, but in general, we're looking up, say, a word and its meaning. In this case, we're going to look up a k-mer and its left and right extension. And the key is whatever that thing is that you're using to look things up. So in a dictionary, it's the word. And the value is whatever thing you need to look up. And in a dictionary, it would be the meaning of that word. So what we're going to do is we're going to come up with a function h that is going to somehow divide the words into different groups so that we have a smaller set of things to look through for the ones that we're looking for. And we're going to store those groups into an array. So one element for each group, and each group will sit there in an array. So that means that given a word or a key, we can look things up, and we do that by applying that function h, that hash function, to the key to see what group it's in, and then we can look very quickly within this group. So let me do it for one really simple example, kind of a trivial example, and then we'll see if this makes sense. So let's say that we are working on the words in the full alphabet, and so we're going to make our hash function h is going to be a really simple hash function, which is just look at the first character and use its numerical value. So a is, what did I do? A is 0, b is 1, and so on. Okay, so really stupid hash function. And now I'm going to make an array of 26 elements. So I've got 26 elements. Each one of them is going to have like a linked list in it or something to store a whole bunch of things, because in a dictionary we're going to have a whole bunch of words that start with the same character. And we can make a, I say 26 squared items. So anyway, so we're going to, we're going to make another table of the, we could also make a table of the 26 elements with the last character. We could also make something that uses the sum of these two, like 26 times the first character plus whatever the last character is, or the second character, for example. So that would allow you to take into account both characters. So now we'd have 26 squared things in the entire dictionary. Okay, so these are some really kind of stupid values of hash functions, but they're ones that make it clear, hopefully, what a hash table is. Once I have decided this, even if you've got really long words in here, we just have to look at, say, 2 characters, and we can store them in a hash table. And now, if you're only looking up, if you're looking this word up in a constant amount of time, I can figure out what the first character and the second character are. I can compute my hash function. So my hash function is just going to be a function of that character and is going to take a constant amount of time. And then add them together. So all of that can be done in a constant amount of time. I'm not reading through the entire word or anything like that. And I can use that then to figure out what bucket it's in, what group it's in. And so I only have to look through all the other words that start with PN in order to find this particular word in the dictionary. Okay. So let me stop there and see if that makes sense. Maybe.
This actually looks like things they've seen before. So bin sorting is a form of hashing, of this kind of hashing, not very smart.
That's right. Yeah, exactly. Right. So bin sorting, if you're going to just sort of put things into buckets first, you do it. So, okay, good. Let's see. What is that saying? Okay, so this is a picture of what my hash table will look like. I've got an array of size k. I'm going to use better hash functions than this, but I'm going to have a linked list of values that are in there. Let's see. So often you have a linked, as I said, a linked list in each one of the elements. This is called chained hashing, and I would say it's typically what we do in most modern hash tables. You might put a tree in the element rather than to make the lookup faster if you happen to get a very large bucket of stuff. A perfect hash function is if you've got a set of keys, it can produce a different table index for each key. So the Kamer example is a good example. If I were actually only doing formers, I could build a table that was big enough to hold every possible former, right, every possible Kamer of length 4, and then I could have a perfect hash function that would just put the thing in the final bucket where it belonged, and there's no issue with two different Kamers hashing to the same place. But in general, I'm not going to be able to have a hash table that big, and therefore I won't have a perfect hash function. So what we'll have is two different keys, two different Kamers, let's say 51 mers, that hash to the same value, so they'll end up in the same bucket even though they're different Kamers. And those are called collisions when two different keys end up in the same bucket. So if you don't have a perfect hash function, that means you have to store the key inside of the table itself so that when you come back later and try to find it, you can say, oh, was that the thing that I was actually looking for, or was it an accidental collision that landed in the same bucket? Yes?
So sorry, is a perfect hash function just a histogram then? Like if you just have like a one-to-one mapping of like possible things and counts?
Yes, yes. If you have a perfect hash function, right, then you may not need to store anything other than the count, if you even care about the count, right? Yeah, right. Yeah, but yes, it would be exactly. Okay. Okay. So let's see. Are there more details in this? I think this was. So in that case, you've got to store the key inside of the data structure so you can look it up later. So here's a little, simple example. I made this back when I was reading Harry Potter all the time, but anyway. This is our dictionary. We've got the names Dudley Dursley, and the value is Muggle, cousin to Harry, and then we've got another thing in the same bucket. So these two, Dudley Dursley and Professor Dumbledore, happen to hash to the same thing. You could see how that might happen if you took a really dumb hash function that says pick the first character of the second word as your hash function, and then they would end up together here. And so then then you can look them up. So that's our picture. Okay. So typically when we're building hash functions, we'll be a little bit smarter than this, and we tend to do things that involve some kind of a function like this. We multiply one of the characters, for example, in the thing that we're trying to hash times some number a, some magic number a, and then we'll add some offset to it. But we might do that for every different character in the set. So I don't want to. There's a whole bunch of literature on what makes good hash functions. You do have to be a little bit careful, and actually there was a study done looking at a whole bunch of compilers for different programming languages out there, and they looked at the hash functions because the compilers inside of them, by the way, use hash functions in order to figure out how to analyze your program text. And some of them had really bad hash functions. And that can happen if, for example, A is a multiple of the size of the hash table, because this function up here, this hash function, is going to produce something that is a big, huge number, and then it's probably much bigger than the size of your hash table. The last thing you'll do is take a modulo operation to get that into something that is a position within the actual array, which is your hash table. And if you aren't careful, you'll end up just statistically ending up with lots of things that end up in the same bucket of the hash table, if that makes sense. Okay. So we'll be careful about that. But that's, I think I skipped that, right. So, you know, so the worst case is still pretty bad in terms of how well does a hash table perform. If we have a really bad hash function that puts everything in one bucket, then we're going to end up with a linked list or something to walk down in terms of finding anything that's in the hash table. That happens if the size of the hash table is too small, or if the hash function is very bad, and then you end up with this linear search problem. One of the things that people talk about in hash tables is the load factor. So what is the load factor of what is the average number of elements per bucket? And you want this to be about 1 because you'd like this to be basically a constant time lookup in the average case. So the analysis that we'll do on hash tables would say in the average case, you've got a good hash function, it takes constant time. You're basically going to hash things and you're going to find it right at the beginning of that linked list, or even if you have to go a couple of links forward in the linked list, you can bound the amount of time it takes. So as long as you don't have either a hash table that's too small or a hash function that's very bad. Okay. Okay, that's for 61B. You don't have a lab this next week. Sorry, I took these slides from a couple different places. So, okay, so that's sort of the idea of how we're going to build the hash table. And let's see, I mentioned something called a Bloom filter. A Bloom filter is an even more clever sort of way of looking at building a hash table. And what it says is, well, I can't really build the hash table I want that's big enough. But it's a statistical data structure that says I'll use a couple of different, let me see if I can do this, a couple of different hash functions. And essentially what I'll do is I will compute a hash on something. I will look to see whether it's in the hash table. In fact, I won't even store like a linked list there. I'll just have more like my histogram, my flat array, and I'll see whether or not the thing is in the hash table. And if it's not there, then I'll go ahead and like mark it as yes, this thing has been seen before, and I'll go on. And if I've seen it before, if I get to that, if I hash the thing and there is a 1 already sitting in that place in the histogram that says, oh, you've seen this before, now go put it into a real hash table. And so this is a way of actually filtering out the k-mers without storing them all and computing the histogram on a whole bunch of things which are erroneous k-mers that only have a count of 1. And so you use this. And that table, that Bloom filter, is kind of tricky because you could accidentally decide that you've seen this k-mer before, right? You hash these two different k-mers, they're completely different. The first one gets written as a +1 in this bucket, the second, but it doesn't get stored. The second one comes along and it says, have I seen it before? And you get fooled into thinking, yes, you've seen it before, so go ahead and put it into the real hash table because it's a good k-mer. And so you got fooled by the Bloom filter. But you'll never get fooled in the other direction, which is if you haven't seen it before, you can be guaranteed if the value in that hash bucket is in that histogram bucket, whatever, is a 0, then you're guaranteed you haven't seen it before. Okay, so that's a way of sort of pre-filtering things. Bloom filters, by the way, also come up all over the place in these sort of distributed data structures that are used in web searching and things like that. So go look at the Wikipedia page if you want to know more about them. But okay. So we're going to run these on one of these big parallel machines at NERSC, and without going into details about this, what I'm going to do is I'm going to take my hash table and I'm going to give each processor a set of the buckets of the hash table. So now what I'm really doing in the implementation is I hash the value and then I send it across the network and the machine and store it in the hash table, or later look things up in the hash table. So let me see. I wanted to go back, I think, to the picture of the what I'm doing in this phase, because I didn't really tell you what that was. Okay, so why am I using this hash table? So I'm using it to try to produce these contigs, and the contigs are, what I'm going to do is take all my k-mers, assume they've been filtered out so all the bad ones are gone. I have only copies of k-mers with a unique left and right extension, so no possibility of saying, oh, it goes two different ways. So I throw all those k-mers into my hash table and with their left and right extensions. And now what I can do is I can pick up an arbitrary one and I can say, oh, add the character in that the hash table says is the right extension, shift my k-mer window over by 1 to the right, because I've now got an extra character on it, hash that value, look it up in the hash table, and if I see another one, then I'm going to take those two k-mers and glue them together and say that's a contig. And I just keep going all the way into the right until I run out of things that I can't find in the hash table, or I go to the left and I also go to the left and try to glue them together. So in that way, I get longer strings of k-mers that are very high confidence, unlike the original reads. We're pretty sure that these are all good contigs, and they're longer than the k-mers. And by the way, I've been a little bit loose about the fact that there's special characters to mark the endpoints and stuff like that of the reads. But, okay.
Are they longer than your original?
They are longer than the original reads. So they might be on the order of 1,000 base pairs, but they also tend to be highly variable. And what it turns out that in the genome, and this is where the JGI people have been giving us kind of a hard time, there's certain parts of the genome they care about a lot, which is the RNA, the part that's coding for RNA. And that has a lot of repeated sequences in it. So those are things that in the actual genome, appear multiple times. And this algorithm cannot resolve them because, remember, this algorithm said if there's a branch, I'm just going to chop it off there and not store a k-mer if I can't figure out what its left or right extension is. And so at this point, we're pretty good at resolving the unrepeated regions, but we can't get through the repeated regions. OK, so the next thing we're going to do is to try to get the repeated regions. We're going to go back and align the so far, we've only been looking at exact string matches of things, right? We're just looking at hash functions. We're making sure that these things are, because we're hashing them, we're looking for identical matches. And now what we're going to do is we're going to do alignment. So we're going to take the original set of reads, which had a bunch of information that we threw out, and we're going to try to figure out if they match up with the contigs that we produced in this previous phase. But we're no longer going to use identical string matching. Now we're going to use something which is genome alignment. And this is probably the last thing I'll actually explain is the algorithm that we're going to use for doing this. So, so what we could do, I will say, is we could take, we're going to run a local alignment algorithm. And in fact, let me do this a little in a different order than the slides are in. So what I want to figure out is whether two strings match with some number of mismatches allowed. So I'm going to allow insertions and deletions and mismatches of characters. And for a given pair of strings, what I want to do is minimize the number of mismatches. So I want to find the alignment that minimizes the number of insertions and deletions and mismatches, flipped characters that have to be changed in order to get those two strings to match. So here's two example strings. And you can see that if I could insert an A let's see, where do I go? An A into the second one. Well, to kind of move that around, actually delete an A and then insert an A, I can get something that matches. There's a T missing on the end. And so, so this is a dynamic programming problem. You probably haven't talked about dynamic programming. OK, so this is a little dynamic programming problem. And the way it's going to work is we've got two strings, and we're trying to compute the best alignment of those two strings with a minimum number of matches. By the way, if the score becomes too bad, we'll just decide they don't really align. But for now, we're just going to say, what is the best possible score we can get for aligning these two strings? So we build a little matrix, and it has the one string going across the top and the other string going across the side. And what we're going to do is, before we start our algorithm, we're going to decide kind of how much we care about insertions and deletions. So we'll say that an insertion or deletion has a penalty of -2. A match has a +1. That means those two strings actually match. And something that the character is just flipped somehow is a -1. OK, so we're going to put zeros in the top row and the side row. And now in each location, we're going to look at the 3 cells that are to the left and diagonally above to the left, and then to the and straight above. OK, so what did we do here? We said, well, the 2 characters I'm looking at above that cell are both a G. So and I didn't say this before, but we're going to take the maximum value that we get by adding these on. Ooh, I didn't really show you the code. I'm going to basically look at either adding a gap penalty if I'm doing an upward moving from the left or moving from above, because that means I took a character from one side, from one string, but I didn't take a character from the other string. So that means I've increased my gap. I've created some space in one of the strings. And then a mismatch, if they're just sort of different characters, I'll assign that a negative. I'll subtract -1 from the score that I'm moving from diagonally. If it's a perfect match, I'll add 1. So up to my diagonally upper left is a 0. There was sort of a perfect match trivially at the beginning of the string, and the first 2 characters match, so I get a 1. And the next characters, so these aren't so interesting, but the next character also is a perfect match. So that gives me, I look up to diagonally up to the left. I add 1 to it because it's another good match. And I think that, oops, oh, and there was another one down here where I said, oh, it's possible that that A actually came from another A. And so then I have to assign a 1 into that cell. Let's see, there it goes again as a 3. And now we had a problem because this one has a C up above and the other one has an A. Sorry, I'm looking at this, yeah, at the 2. So I had to subtract 1 from it because there was a mismatch there. Okay, and this goes on, and we run this algorithm, and I'll just show you the sort of line that you get through here. And this turns out you can follow this path back. You look for the maximum value in this table, and you follow this path back. You've kind of recorded how you got there, and you'll go back to, you'll find the actual string. And the reason there's a jog in this is because that's a place where you had to insert something in order to make it past that part of the table. Okay, so this is called the Smith-Waterman algorithm, and this is what's used kind of down in the bottom to figure out whether 2 strings align. This is very expensive. You do not want to do this for every possible pair of reads and reads and contigs that you've computed. And so what you actually do is, shoot, this is kind of slow, but you use an algorithm called a seed and extend algorithm. Let's see if I can go backwards more quickly here. So this seed and extend algorithm, what I'm going to do is I'm going to try to figure out, is there a high probability that a given read, the red and blue stuff up there, matches the green, the contigs. So what I do is I go through my contigs, I compute k-mers again, I stick all those k-mers into it for my contigs, I stick all those k-mers into a hash table, and I say, well, those are all little fragments of the DNA that are in there. And I'm going to say, if I can't find an exact match with a k-mer inside that contig, I will not try to align this read with this k-mer. Okay, so now you can use a different k-mer length at this point, but this is, again, it's making sort of a statistical assumption that if that k-mer length is short enough, I'm guaranteed that at least 1 k-mer between any read and any contig will match, and so therefore I'll run the expensive alignment algorithm on them. If no k-mers match, then I'll kind of, I'll assume that, that they don't align. Now, you could be wrong, right? If you have badly spaced errors that just hit your k-mer length, you're using a k-mer length of 51, and you've got these k-mers that exactly every 51 characters, you'll never find a common k-mer. But since that has a low probability of happening, this way of storing these k-mers and then only computing the expensive thing on pairs of ones that have one substring that has an exact match is a way of cutting down on the complexity. And that, by the way, this algorithm comes up all over in other computational biology problems, not just in this particular case. So let's see, any questions about either alignment or, I also should say alignment, if you remember that pie chart from the very beginning, is what JGI actually spends the bulk of their computing doing. Because a lot of times what you're doing is you have some reference database of, for example, all of the microbes that you've computed that you found in all of history in your database, and then you say, I've got some new DNA, and I want to just align those reads against the things that are in the database. And you either can try to assemble it first and then align it, or you can just try to align it. So you're doing alignment typically many times. And you also do alignment on genes to try to figure out whether or not they're similar to each other and stuff like that. Okay, so we're building another hash table. In this case, the hash table is really the contigs. The contigs are the keys. Sorry, oh, this is step 4. So there's a little bit more work that goes on in here, and I don't really have time to talk about it. I'll just say you go through and run kind of a little local assembly in any place where there's some space filled in, and you take the reads that fit in that gap, and you go back and try to fill it in. Okay, so let me just sort of say what, when I think about both data structures, and in this case I was thinking about what hardware you want to run these kinds of algorithms, it's sort of hash tables all the way down. So I told you in some detail about a couple of them, histograms, hash tables. There were 2 that we talked about, and there was another one that I didn't really talk about. So ideally what you have is just this big, huge shared memory machine, because these hash tables are things where you just want to kind of write into any location in memory. So you run a really big memory that you can access every bit of memory really quickly, and unfortunately you can't really build those machines. So we have to have some other kinds of hardware. So I think at that I will not show you all the performance results here, except to say, wait, where's my summary? Yeah, so it's got, I think that people have sort of assumed that you have to run bioinformatics problems on these kind of shared memory machines. I didn't talk about the parallelization, but we do parallelize this code, and this looks quite different than the kinds of things that you do in physical modeling and simulation, which I think has been the bulk of the examples in this class. The assembly problem is only one problem of many, and in fact is not the dominant part of the JGI workload, but alignment comes up all over the place. And this sort of seed and extend algorithm that I talked about, I said you want to align a whole bunch of things to a whole bunch of things. Well, what you'll do is you'll hash some subset of them, substrings, so that you can figure out which things to actually run the expensive algorithm on. And there's other data structures that come up in here, trees, phylogeny trees, and things like that, and tries, which are data trees for storing strings of things. But these tend to look like a lot of strings and a lot of hashing, and a little bit different than the other things. So I'm happy to take any other high-level questions or other questions. Phil.
So this has gone in the last 20 years, I guess, from something that was really hard and really expensive to something that's really cheap. What's kind of the balance of hardware, algorithm, software in that kind of progression?
Yeah, so Phil's question was, I guess 20 years ago it was really hard and expensive, and now it's really cheap. So do you mean the sequencing?
Yes.
Yeah, so the sequencing has gotten really cheap, and in fact, a lot of times if you look at articles that talk about the cost of sequencing the human genome, what people are worried about is the cost of sequencing is down under $1,000, and that's for the chemicals, and kind of if you amortize the cost of these big, huge machines over thousands of runs or millions of runs or whatever, then the cost is $1,000. But the cost of computing and analyzing that genome has not necessarily gone down as much. And part of it, what we're doing is trying to say, well, actually, if you use really clever algorithms, and you probably use parallel machines, that's not to save on money, but that's to shrink the total time it takes to run these experiments, then you can actually bring that number down. For some of the genomes, actually I heard a great sort of anecdote. There was a Nature paper published recently on 150 human DNA, which they did de novo assembly on, so not using a reference. They did de novo assembly on for 150 people in Denmark. It took them 10 years to analyze that data. And they published the paper recently. And they said probably it took them like 9 years to do the first 2, and then the last 148 were much faster. But we could do this in parallel on a parallel machine in about 4 minutes per DNA, so we could do the whole thing in about an hour on one of our parallel machines. But people are still kind of trying to figure out how to use these parallel machines. But there are other examples, and I didn't talk about the protein clustering, which Aiden may mention. But in that case, and in some of these big metagenome assemblies, they run them for many days or even a couple of weeks, and then sometimes they crash and run out of memory because they run out of memory. So that's sort of a typical behavior. And some of them are very fast, and you can do it on any old workstation, and it's just fine. But some of them are very expensive, and it has to do with the complexity of the microbiome, the metagenome, as opposed to necessarily even the size of the input data that you're working with. And so, yeah, it's kind of all over the place. But some of these problems, people are running them for weeks in some cases. Any other question back, and then one more.
If you had a soil sample and you had 2 species in it that you had never seen before, how does it know that they're different?
So that's a question I try to defer to my biologist colleagues, but there's sort of some rules that they use for figuring out whether these are 2 independent species or just 2 different variations. So actually another person we were talking to, the one who talked about this Denmark study, is doing a big pangenome study. So that looks at the strains within a species, and you're trying, in that case, you don't want to just get strings out. You want to get out the graph that says, here's the common parts of the DNA, and then here's all the different versions that we happen to see inside of that genome. And we don't have our code really set up to keep track of that, but you can imagine if we kept a lot more information around, we could produce a graph that said, here are all the different paths through this genome mess. And I don't know what and I think there's actually some controversy about when is it a new species and when is it a strain of an old species. And that's sort of, it's not my job to figure that out, but somebody has to. Yeah. One more question.
So you kind of described how you generate these contigs, and you talked about how you kind of like window assembly. Do you do this like in parallel? So like you're like multiple processors are trying to develop these, and if you do that, how do you know like this processor did start in a different place and like walk back and like you're doing the same work twice.
That's why there was a PhD thesis in this, among other things. So yes, that we build this big distributed hash table. Each processor has a subset of the buckets. Every processor picks up some k-mer and starts walking left and right, and they can run into another processor that's working on the same things. So you mark them when you go by them, but if you sort of really actively get to the same place and you're both trying to go in the opposite direction, then there's a little protocol in there that says, you know, you take it, no, you take it, no, you take it. You know, so that really that's exactly what happens, and then eventually it gets resolved, and one of the processors goes forward, and the other goes and picks up another random k-mer and starts from scratch. Okay.
Any other questions? Well, let's thank Kathy.
Thank you.
Programming assignment 5 is out. Scheduled for the next